Now we're going to look in more detail at some of the bacterial structures like the cell envelope and the internal structure of bacteria. And very briefly, we'll compare with Archaea. Most bacteria have a cell envelope outside the cytoplasm, and it's got three basic layers. You've got the cell wall, the cytoplasmic membrane, and then some bacteria also have the outer membrane. So those two basic categories of bacteria that I've spoken a little bit about that are really, really important are going to be gram-positive and gram-negative. We categorize most bacteria gram-positive or gram-negative, and this is based on differences in the cell wall. Okay. Gram-positive is going to be shown here on the left, gram-negative here on the right. Gram-positive, let's see, well, the cell wall, first off, is going to be made of peptidoglycan, okay? So you're going to have the cytoplasmic membrane here on both, all right? And then you're going to have this really large layer or several layers in gram-positive of peptidoglycan, and we're going to have some teichoic acid in here and some lipoteichoic acid in here. And you're probably thinking, why on earth do I care about the cell wall of bacteria? Well, you care a lot because, one, it helps us classify them. It helps us to identify how virulent they are, partially anyway, because gram-negative are in general much more virulent than gram-positive. It is how we stain them to identify them is based on the differences in the cell wall. And not only that, and I may have already said this, but if you want to break into a bank, I'm pretty sure I said this, you want to break into a bank, what's the first thing you're going to do? You're going to look at the floor plan. You got to know how you can break into something. So if you want to kill these bacteria, you got to figure out how to get into their little cell, okay, in order to kill them, all right? And so this is a really good diagram here. I'm not sure I used the same one when we get into more details about this, but you may want to come back and look at this graphic. I'm pretty sure this has come from your book. You want to remember this is much, much thicker in gram-positive, but there's only this one layer of cytoplasmic membrane. And in gram-negative, the peptidoglycan is very thin, but you have an extra outer membrane layer. And so this is why when we say gram-positive or gram-negative, we're talking about a staining protocol. And this is why one will stain a purple color and the other will stain a pink color, okay? And it's because of the differences in the cell wall. And it also, like I said, plays a role in how we are able to treat them. The cell wall is very important to us. It gives bacteria several important characteristics. It helps provide shape, support, and it prevents the bacterium from undergoing cell lysis. And so we have certain drugs that help target the cell wall and disrupting the cell wall. So if we want to break in, we have to try to get through that cell wall. Okay, so that's one of the main ways that we do it. And so we can disrupt or cause problems in the integrity of that wall and cause cell lysis. So lysis, you want to think loosen, which basically means we can cause that cell to rupture. It's kind of like popping a balloon, okay? And so if it bursts, then we have basically killed the bacterium, okay? And remember, it gets the strength from this peptidoglycan. Bacteria live in an aqueous, usually watery environment, and in their cell is going to be a water-based or aqueous environment. Water is going to be absorbed by them through osmosis, the process of osmosis, and the strength of the peptidoglycan in that cell wall prevents them from rupturing due to, as I've already said, cell lysis due to osmotic pressure, which means taking in too much water, okay? So if you think back to biology, we had cells without a cell wall and cells with a cell wall. Cells with a cell wall are much less likely to rupture because they have an extra layer of reinforcement, okay? So it's important to us, though, because how our antibacterial drugs work. Medications like penicillin or cephalosporin target the peptide crosslinks. Okay, and we're going to look at that structure in the peptidoglycan, which basically dissolves that cell wall. So we've taken that outer layer of protection away, so the bacteria can undergo cytolysis. They can take in too much water and rupture, and so they're not able to survive. Okay, good for us. Our bodies also have a natural antibacterial substance, which is lysozyme. And the lysozyme is found in tears and other things, saliva, that hydrolyzes. Hydro means water. Lysis, remember, loosening. So water loosening. So basically, again, breaking down that cell wall. All right. Hans Gram developed the Gram staining technique, which is where we get two different colors. We can differentiate between these two main categories of bacteria by undergoing this particular staining method, which you are definitely going to need to know every step of and why each step does what it does, which we're going to go through here in a minute. So he developed this technique that identifies the two major classes of bacteria as gram-positive, which is going to be purple, and gram-negative, which is going to be pink, based on the different construction of their cell wall and the chemicals we use to do that. And so it's kind of tricky to remember which is positive and which is negative, because pink and purple are both P's, but you need to get that straight in your mind now. Okay, you have got to know the difference between gram-positive being purple, gram-negative being pink. Okay, let's look at these differences. All right, so the gram-positive cell wall has a thicker wall, multiple layers of that peptidoglycan, so 20 to 80 nanometers in thickness. Important to remember, while that layer is much, much thicker, it is only one layer. It is also important to remember that it contains the teichoic and lipoteichoic acid. Okay, these are kind of like the little, if you want to think of these as Lincoln logs, okay. This is the peptidoglycan here running this way, and the teichoic acid is what holds the strands together this way, okay? And the teichoic acid is a polymer of alcohols and phosphates. Now, gram-negative cell walls have that thinner sheet here of peptidoglycan, but remember they have this second outer membrane, and then there is what we call the periplasmic space between those two. The outer portion is very important, this periplasmic, this outer lipid membrane here. It's important to its virulence, because when the bacteria die, it sheds this off, and this is going to be endotoxin. That lipopolysaccharide here is going to create a toxin that can cause symptoms even after we kill the bacteria, which is really important to understand, and I will probably harp on that all semester long, okay? So it's a lipopolysaccharide. It acts as an endotoxin, and we're also going to look at exotoxins, and so to me it's a little counterintuitive because it's breaking off. When you kill the bacteria, this breaks off. I always want to think exit, but it's backwards from that. It's endo. It's within the bacterium itself and not produced by the bacterium. I guess is the way to keep that straight in your head. But the endotoxin, and you get that when you kill the bacterium, so the bacterium does not even have to be present anymore. So like in food poisoning and botulism, the bacteria can be removed, but this toxin can still be there, okay? And so it can still give you a toxicity, all right? It's going to cause fever and shock with a gram-negative infection. That's going to be things like meningitis or typhoid fever. Gram staining is, if not the most, one of the most, and probably is the most important procedures that we do in lab, okay, other than learning aseptic techniques. You're going to need to know all the steps, why they do, what they affect, and when it's gram-positive or when it's a gram-negative bacterium. Okay, I'm sorry, my phone's going off. So let's see. Once the bacteria are heat fixed, which means we've put them on the slide and we've waved them through the flame a few times to kind of get them to stick, then we're going to go through these steps, okay? And you will do this in lab. So you're going to drop 5 or so drops of crystal violet, which is a purple dye, on the smear, and that was going to stain the bacteria purple, okay? So at this point, and you also need to know that at each step, what are things going to look like? So at this point, everything's going to look purple. So gram-positive and gram-negative are both going to look purple. One thing that's not listed here in the text is you need to rinse after. Okay, if you don't rinse after, and you may not see this in the virtual lab, but in real life, we get these big clobs of crystal violet and iodine crystals, and it's useless. You can't see anything. Okay, so rinsing is really very important. And so then we're going to apply 5 drops of grams iodine to the smear. We're going to let it sit for 30 to 60 seconds. And everything you kind of let sit for 30 to 60 seconds except for one. You're going to rinse with water after that. Okay. Iodine is actually a stabilizer, or what we call a mordant, in the gram-positive cell walls, but not the gram-negative. Okay, so it's going to stick. This purple color is going to stay. This purple color is going to stay with the addition of the mordant on the gram-positive cell walls. It gets trapped in the wall. Okay, and so that iodine's not going to make it stick in the gram-negative bacteria, though. So we're going to then decolorize. That is our third step. We're going to add alcohol. Okay, alcohol is going to actually dissolve that outer membrane in gram-negative bacteria, and so then the purple color goes away. Okay, so we get rid of the outer membrane, the purple color goes away. Okay, and so the bacteria are no longer going to be purple. We're going to have purple, and we're going to have clear for gram-negative. So then we're going to add a second dye, and this is what we would call a differential stain, because we're getting 2 different results. We're going to add or drop safranin, which is a red dye, onto the smear, let it stand for 60 seconds, and then rinse it with water. Okay, the only thing you don't let stand, I'm sorry, is the alcohol. You put it on and then rinse it off immediately. And so you're going to look under the microscope then. After you rinsed this off, you're going to blot it dry, and you're going to look at it under the microscope, under oil. Okay, and so what you're going to see is, if it is gram-positive, it will be purple. If it is gram-negative, it'll be pink to red. Sometimes it is kind of difficult to tell what the color is. Is it pink or is it purple? Because it kind of can be in between. And sometimes you have more than one bacteria, and you actually have both colors. Okay, you must know all these steps, what it looks like at each step, what it does to the cell wall, and why. Okay, really important, this particular slide right here, both lab and lecture. If you've had me before, you also have probably heard me say, maybe not as much as I used to, but there is an exception to every rule. I used to say that a lot in chemistry. Always an exception to every rule. So some bacteria are what we call acid fast. They are not typical. So they have a different type of cell wall. And so we use another method. Okay, acid fast will stain red if the bacterium are acid fast. Okay, so we have things like Mycobacterium that are acid fast. Remember, that is when you see myco, you need to be thinking fungi. They contain peptidoglycan, and they can actually stain positive, but the bulk of their cell wall is actually made of unique lipids. And so you can actually use this as a second test in some cases to differentiate these. And so acid fast is going to stain red. Part of this is because it has this mycolic acid. Archaeans are, of course, different, and so they have some non-typical or atypical cell walls. They're made completely of polysaccharides. There are some that are made completely of protein. None of them have actual peptidoglycan. They may have some peptidoglycan-like substances, but they're not made of peptidoglycan. And then some don't even have a cell wall. Here are some other exceptions. We have mycoplasmas. These have no cell wall. They have sterols, which are cholesterols, in the cell membrane that have to stabilize the cell against lysis, or having that osmotic pressure cause them to rupture. An example of this is Mycoplasma pneumoniae, and this is the bacterium that would cause what we call walking pneumonia, which is a milder form of pneumonia. And then we have L forms, and these have a cell wall, and then they actually shed their cell wall as part of their life cycle later on. And so these are often responsible for persistent infections, which means those infections that don't want to go away, and they can also be antibiotic resistant. The outer membrane of a gram-negative bacteria contributes to an extra barrier, so it's a little more virulent. So it contributes to making it more difficult to inhibit or kill. Okay, so it's more resistant to certain antimicrobial drugs. Alcohol-based compounds can actually dissolve those lipids or fats in the outer cell membrane and damage the cell. So alcohol swabs used to cleanse the skin are used to cleanse the skin before certain medical procedures. And then treatments of infections caused by gram-negative bacteria require drugs that actually can cross the outer membrane, okay? And so this is some of the important points of those differences in cell wall structures when we look at clinically applying this information. Water is a solvent for all cell contents, basically, and so it's going to contain sugar, amino acids, and salts in it. But the cytoplasm is going to be about 70 to 80% water, and then 20 to 30% the rest of those substances. And so basically it serves as a pool or a holding place for the building blocks for all of the cell synthesis, for energy. When you hear the word cytosol, that's referring to the liquid portion, but we're not really going to distinguish between the two. So bacteria, remember, can have these plasmids, this circular DNA that's actually separate from the main DNA. And this can house hereditary material, non-essential portions of the DNA. It can pass along drug resistance. It can pass along new toxins, and it can pass along the ability to produce certain enzymes. And so this can enhance other bacteria. So basically, it's like going to somebody and learning how to do a new trick, okay? Which is not good for us, but good for the bacteria. Ribosomes are important to us because ribosomes are the place where protein synthesis occurs, okay? Proteins are basically the foundation for almost everything in a cell. And if we can stop protein synthesis, we can stop the bacteria in its tracks because it can no longer replicate. Okay, so this is why we care about ribosomes. So ribosomes, you'll have a ribosomal unit here. You'll have a large unit and a small unit, okay? And these are where we make our protein synthesis. They are going to be 70S for archaea and bacteria. For eukaryotes, we have ribosomes as well. They're just 80S. So just know that's kind of a little bit of the difference between their ribosomes and our ribosomes. Bacterial endospores are really good at helping bacteria adjust to changing and difficult or extreme habitats. And so they are protective in nature. And again, they contribute to the virulence of a bacterium. The endospore is the best at ensuring survival in a tough condition. Things like Bacillus clostridia or Spores archaea are able to form these endospores, and it's basically like where you have a two-stage life cycle. You have what we call a vegetative state and then the endospore. Now, vegetative state, you're probably thinking like a person that's in a vegetative state that's not doing anything. That's not what we mean here, okay? This is a metabolically active state. This is where we're undergoing growth and reproduction. We're normal cellular activities, okay? So when the environment gets harsh, the bacterium can undergo sporulation, and that's going to cause the bacteria to form this spore, this endospore, and protect the bacteria from these harsh conditions. So it's almost kind of like hibernating, okay? And so they don't usually reproduce during this time. They can resist extreme heat, drying, freezing, radiation, or other chemicals that normally kill the cell. So it makes them very hard to kill. So normally we heat things up to kill bacteria, right? Well, if it has endospores, it's not getting up, okay? So really important here. And I know I say really important a lot. So just to give you an idea of some of the things that endospores help contribute to these particular bacterium for persistence and resistance in infections. Persistence means being able to stay there and you can't treat it, can't get rid of it, so it sticks with you a long time. Resistance means that you have a very difficult time treating it with certain antibiotics or antimicrobials, okay? So endospores inhabit the soil and dust. That's where you'll typically find a lot of these. They're constantly issues where we have sterility or cleanliness as a factor, okay? They are resistant to normal cleaning methods like boiling water, soap, or disinfectants. This is a list of 5 of these, and you've probably heard of most of these diseases that they cause if you don't know the names of the bacteria. Bacillus anthracis, which is anthrax, okay? And we're going to study anthrax later on. You'll hear that there's 3 forms, okay? Pretty nasty stuff. Clostridium tetani is the cause of tetanus. Well, most people think tetanus, they think lockjaw, and so they think it's not really that bad. Well, I hope to change your mind on that. I'm going to show you a picture of tetanus that will be eye-opening, okay? Clostridium perfringens, this is what causes gas gangrene. So think Civil War soldiers getting their legs cut off because they had a few cuts in their legs. All right, not good. And then botulism, which is basically food poisoning that kills you. Okay, and you can't see it, you can't smell it, and it's the endotoxin that gets you. So even if the Clostridium botulinum is removed, you can still get sick and die. And this is actually the component of Botox. So I hate to break it to you, but if you are undergoing Botox injections, you're putting botulism in your face. Okay, and you'll notice 4 out of these 5 are Clostridium, okay? The last one is Clostridium or C. diff, which is a very serious gastrointestinal disease. And you're thinking, diarrhea, how bad can it be? Well, it can be life-threatening, okay? This can get into your intestinal wall and sit there and just destroy your tissue. And it is a profuse watery diarrhea, so very, very dehydrating. All right, let's see if you can apply some of what we have discussed to some of these practice NCLEX questions. So when you're trying to pick your answer, think about what we have talked about, and that will help you, okay? So why is an alcohol swab applied to the skin as antiseptic prior to getting an injection or a shot? All right, so our choices here are alcohol helps stabilize cytoplasmic cell membranes. Alcohol swabs prevent infection by dissolving the bacterial lipid membrane. Alcohol inhibits protein synthesis in gram-positive bacteria, which is most commonly present on the skin. Or rubbing alcohol, the rubbing action congeals the cytoplasmic membrane. So this is all stuff we've talked about. So I want you to pause and see if you can get the correct answer. Okay, so if you picked B, that we dissolve that bacterial lipid membrane, you're correct. If we dissolve that membrane, we make that bacteria more susceptible to cytolysis and our own host immunity. We can now go in and kill the bacteria. So it's B. All right, winding down this chapter 3 here, we've got Archaea. Remember our other prokaryotes. These are unusual cells. They're single-celled and they're pretty simple. They do share some characteristics with bacteria, but they are actually more closely related to eukaryotes if you look at their RNA, your ribosomal RNA sequences, okay? They have similar ribosomal, or actually some that are the same RNA sequences, and the ribosomal subunits are similar. So how are archaea different from other cells? They're different because they do have the unique RNA sequences. Their DNA collects or compacts differently than other mechanisms of making the DNA smaller, and they have unique membrane lipids. They have some really interesting critters here, though, okay? They have what we call extremophiles. Phile means loving, okay? So they love extreme conditions. And when I was in high school, and in fact, it hasn't even been that long ago, it's just been, I don't know, a decade or two, we have discovered that where we once thought nothing could live, that we actually have life. We have these archaeans, okay? So in these deep sea hydrothermal vents, all right, like where you have hot lava, you would think anybody could live there, right? Well, you could have one of these extremophiles, a hyperthermophile, a heat-loving microbe that can actually survive there. You can have methanogens, which actually convert carbon dioxide and hydrogen gas into methane, okay? You can have halophiles, which are salt loving. That's where the saline is coming from. You ever heard of the Dead Sea? It was called the Dead Sea because they thought nothing was living there. Well, you know what? Halophiles live there, okay? We can also have psychrophiles, which also like the very, very cold temperatures. So you would believe in the Arctic, in the Arctic especially, that there would be nothing living. Well, you know what? There are some that are living, okay? Some do live on the human body, and they do have the potential to cause disease, but we don't know of any that actually cause disease, okay? So that comes with that kind of little, they could, but they really haven't yet. This is a really good review slide comparing and contrasting bacteria, archaea, and eukarya. I would highly recommend actually making your own compare chart here and put the similarities and differences on them using your textbook. Then come back and check your list with this. Because if you go with your textbook, if you go through your textbook and actually look up and find these differences yourselves, you're going to actually know most of the information by the time you get done making the chart, which is much easier than going through and trying to memorize a chart somebody else has made. If you listen to some of these study tips I'm giving you, you'll find that you spend less time studying, okay? And you actually learn the material for the long haul, which is our goal, because one, you got to pass the NCLEX test, right? And two, or other tests, you need to know this information for your job. So you want to know what you're doing, don't you? All right, so what are species, subspecies, and serotypes? And we're going to talk about this. So this is just kind of a little catch-all for some bacterial terms, vocabulary terms here, okay? So a bacterial species is a collection of all bacterial cells which have a similar overall pattern of traits, okay? And so they should share at least 70 to 80% of their genome. You will often see things like SPP or SP after the genus name, and that means all the species, okay? So don't be thrown off when you see that. A subspecies or strain is going to have the same species with a little bit different characteristics, kind of like our coronavirus that we are currently experiencing is a new strain, so it has got different characteristics than the original coronaviruses. And then we have serotypes, which are portions of a species that give us a very distinct antibody response because they have a unique particular arrangement of serum of surface molecules that our blood serum reacts to.